Hey guys, Aruj here, and today we're talking about protein synthesis. There's actually two steps to protein synthesis, to how we make a protein. There's transcription and translation, and today all we're going to talk about is just the first part, transcription. The next lesson we'll look at translation. Now, all the information to make you is stored in this molecule called DNA. That information to make you gets transferred, it gets rewritten, gets a copy made into this molecule called RNA, and then RNA is going to be used to make a protein. And ultimately, you are made up of thousands of different types of protein. So how do we get to you? Well, it goes information in DNA, transferred to RNA. RNA is used to make a protein, and all of those proteins make you. And we call this the flow of genetic information. That's called the flow, okay? The information starts here at DNA, and the end result is a protein, and then all the proteins ultimately make you. Now, this concept of DNA, RNA, and protein, sometimes that's called the central dogma. And what that means is it's like saying this is the foundational rule. This is the foundational rule for all life. And they say that because all life runs on this, this method of information, DNA to RNA to protein to that organism. Okay, so all life runs off this, and that's why we call this the central dogma. Or it can be referred to as the flow of information, of genetic information. So again, like I said, protein synthesis is broken down into two steps, transcription. Now, when you hear that word transcription, or to transcribe something, what that means is to write, and that's exactly what I want you to think about. Transcription, we are writing something. The second part is translation, and it's just like how it sounds. To translate is to interpret something or to read something. That's what's going to happen in the second part, translation. So if we were to look at where exactly does this occur at in a cell, well, transcription is going to be copying DNA, and that's going to be in the nucleus. So all of transcription takes place here. Then we go to the cytoplasm. And in the cytoplasm, we find a ribosome. A ribosome is going to help us assemble the protein that we need. And again, transcription is in the nucleus, translation is in the cytoplasm at a ribosome, okay? Now, here's what transcription actually looks like. It's kind of similar to DNA replication. So we have our double strand of DNA, and again, we're in the nucleus. We're going to have a new enzyme called RNA polymerase. RNA polymerase is going to do two things. It's going to split open the double strand, and then it's going to copy a molecule of messenger RNA, of mRNA. Now, whatever this bottom strand of DNA says, whatever that bottom strand says, that's what RNA polymerase is going to add to it. However, instead of thymine, since this is an RNA molecule, instead of thymine, we're going to have uracil. Okay? So if there's a thymine in DNA, we could still have an adenine in RNA. However, if there's an adenine in DNA, that thymine is going to be substituted with a uracil. Okay? So RNA polymerase makes this molecule that we call messenger RNA, and then messenger RNA simply just leaves the nucleus and goes to the cytoplasm. Now, why does this happen? Well, DNA is a little bit too big to leave the nucleus, and you actually don't want your DNA to leave the nucleus because it's safe there. So instead it makes a copy, and that copy is allowed to leave. And that copy is called mRNA. So if we were to do a quick recap, the first thing we talked about, the flow of genetic information, which is also called the central dogma. The information starts in DNA, it gets transferred or transcribed into RNA, and then we use that information to make a protein, and you are made up of proteins. Protein synthesis has two steps. Transcription, which means to write. To transcribe is to write. That takes place in the nucleus. Translation, which is to interpret or read, we have not talked about that yet. That takes place in the cytoplasm at a ribosome. In transcription, RNA polymerase uses DNA as a template to make messenger RNA. Now the base pairing, we do have cytosine and guanine pairing together, But anytime there's an adenine in DNA, we're going to have a uracil in RNA. Okay, A pairs with U. And one more thing I forgot to mention. Every 3 letters, let me go back real quick. Every 3 letters in messenger RNA, that has a special name, and you'll hear this name more later. That name is called a codon. Okay? Every 3 letters in messenger RNA is called a codon. Okay, go ahead and get this section in your notes. This will be the main notes. If you have any diagrams or anything extra, that's totally fine. And that was it. In the next one, we will cover translation. And I hope that was helpful.